Hello everyone, let's get started. Today we're going to be picking up our discussion of cellular respiration by discussing oxidative phosphorylation and fermentation. So if you recall, in the last lecture, we talked about the long, complex process of cellular respiration, the process that begins with glycolysis and ends with electron transport. At the end of the last lecture, we talked about how as electrons are passed down the electron transport chain and accepted by the final electron acceptor, the energy from those electrons is harvested and used to build a proton gradient. In eukaryotic cells, that gradient builds up in the intermembrane space in the mitochondria. We said that the next stage in this metabolic process is oxidative phosphorylation. And oxidative phosphorylation is going to use the energy from that proton gradient to build ATP. So, as a reminder, we are still in the mitochondria at this point, and we are looking at a critical enzyme called ATP synthase, which is embedded in the inner mitochondrial membrane, near where the electron transport chain is also embedded. ATP synthase is a fascinating enzyme. It's actually found in all living cells on planet Earth. Obviously, only eukaryotic cells have mitochondria, so in cells that are prokaryotic, in organisms that are found in the bacteria and archaea domains, the ATP synthase molecule is located in the cell membrane, not the mitochondrial membrane. But it is there to do the same work. It's there to build ATP with oxidative phosphorylation. ATP synthase is actually an enormous protein. It's what we refer to as an enzyme complex, and it has two distinct subunits in it that are connected by what is best described as a stalk. The catalytic portion of ATP synthase is the F1 subunit, and this is the portion of the molecule that is suspended below the membrane. The other part of the ATP synthase molecule is called the F0 unit, or F0, which is pronounced F0. This is the membrane-bound portion of the molecule, and this is the subunit that has a tunnel within it, a channel within it. So the hydrogen ions that have been built up inside the intermembrane space are going to flow back into the matrix of the mitochondria through the F0 subunit, through that channel in the membrane. And as they do that, as they flow, the energy that is stored in this gradient is going to be transformed into kinetic energy. It's going to cause that stalk to spin the F1 subunit, to literally physically rotate the F1 subunit. The F1, as it is rotating, as it is spinning, changes shape slightly. And that shape change is going to catalyze the phosphorylation of ADP to ATP. So it's a long, complicated series of steps to go from the hydrogen ion gradient to the formation of ATP through this important molecule called ATP synthase. There's the transformation of energy going on here. The energy that's stored in that ion gradient and the motion energy, the kinetic energy in that F1 subunit that is being used to build ATP through oxidative phosphorylation. On this slide, you can see a drawing of ATP synthase down at the bottom. And up above that, you're looking at an electron micrograph that actually shows what ATP synthase looks like. They're describing it as having a lollipop-shaped stalk, and then that knob portion, the F1 portion, hanging below the membrane. This enzyme, ATP synthase, has been studied extensively, as you can imagine. It's such an important molecule, and again, it's found in all living cells. Down at the bottom here, we can see it drawn out diagrammatically. You're looking at the interior region of a mitochondrion. This blue area up here represents the intermembrane space. So again, the outer mitochondrial membrane would be located outside of this picture. Here's the intermembrane space in blue, and here's the inner mitochondrial membrane. And finally, this represents the matrix. So ATP synthase is shown in green, and the F0 subunit is embedded in the membrane. This is what we call the stalk, and down here is that lollipop-shaped F1 subunit. So this is the ion gradient that we're going to make use of. This is the ion gradient that has potential energy. All of these hydrogen ions in this gradient are going to be allowed to flow back into the matrix through a tunnel in the center of the F0 subunit. And as they flow, their energy, their potential energy, is going to be transformed or converted into kinetic energy. The stalk is going to start to spin, and that in turn will spin the F1 unit, causing it to change shape very subtly. And the energy will then be used to take ADP and phosphate and build ATP. Remember, oxidative phosphorylation is an extremely efficient process. We're going to build a lot of ATP, relatively speaking, using this enzyme, this ATP synthase enzyme, and the energy in the proton gradient. This is a diagram that you've seen before. We looked at it, at least a portion of it, when we were discussing cellular respiration. So remember, this is a diagram that represents the mitochondria over here on the right, and the cytoplasm in the cell over here on the left. And we looked at the portion that summarizes cellular respiration in the last lecture. We looked at how glucose is converted to pyruvate through the process of glycolysis out here in the cytoplasm, and then how pyruvate gets transported into the matrix of the mitochondria, where it is converted to acetyl-CoA during pyruvate processing, and then the acetyl-CoA is converted to carbon dioxide through the Krebs cycle. Now, if you notice, some other parts have been added to our diagram. Number one, we can see the electron transport chain now, embedded in a region of the inner mitochondrial membrane. And we can see ATP synthase embedded nearby in that same membrane. So now with this diagram, we can walk all the way through all of the many processes that make up cellular respiration and oxidative phosphorylation. Now, you'll notice that at the end of this, they talk about 26 ATP, and I want you to ignore that number. The reason I want you to ignore it is because this number depends on what organism you're talking about. When cellular respiration and oxidative phosphorylation occurs in an aerobic organism, in other words, in an organism that is going to put oxygen at the end of its electron transport chain, you get a particular number of ATP, and it's a high number of ATP. Remember, though, that not all organisms do that reaction, because not all organisms are aerobic, number one, and there are organisms that don't do cellular respiration at all. So, remember, there are some organisms that are going to do this process, cellular respiration and oxidative phosphorylation, right here, but they're not going to use oxygen at the end of their electron transport chain. There are microbial organisms, bacteria and archaea and the like, that are going to use a different molecule in place of oxygen here. They're still going to make a lot of ATP, but it's not going to be the same number that is made when oxygen is that molecule. Remember, too, that not all organisms do this. There are some organisms that use fermentation instead. So this number really varies. In general, I tell students somewhere between 30 and 36 molecules of ATP will get produced for each mole of glucose that enters in. It's possible for organisms to do cellular respiration without oxygen. You and I and other animals do cellular respiration in the presence of oxygen. We are aerobes. Oxygen is the molecule that sits at the end of the electron transport chain and acts as the final electron acceptor. But there are many bacteria and archaeal species who use final electron acceptors that are not oxygen. For example, some of them use nitrate, some of them use sulfate, and even carbonate as a final electron acceptor. These organisms are doing anaerobic cellular respiration and oxidative phosphorylation. They're doing it without oxygen being involved. What's important to know is this: they're still using an electron transport chain. They are still using ATP synthase to make ATP. So they are still doing oxidative phosphorylation. That word oxidative does not mean oxygen. It means oxidation reduction reactions are involved. Now I'd like to switch gears here and start talking about a completely different metabolic reaction called fermentation. When human beings and other animals who perform aerobic cellular respiration and oxidative phosphorylation, when they find themselves without oxygen, and when other organisms who use another final electron acceptor find themselves without that electron acceptor available, the process that breaks down the glucose molecule is still going on. Electrons are still being released from glucose, and electron carriers like NAD+ are still being reduced to NADH. But suddenly the NADH has nowhere to go. Remember, normally NADH would become oxidized when it donates the electrons it's carrying to the electron transport chain. But the electron transport chain can only function if there's a final electron acceptor molecule at the bottom of it. So, when the final electron acceptor molecule is not available, the electron transport chain stops. And what that means is suddenly any NAD plus that was available in the cell will quickly become NADH because the earlier processes are still going on. But the NADH is stopped in its tracks from donating its electrons to the electron transport chain because the electron transport chain closes down. This is a life-threatening problem because with the electron transport chain closed down, that means no proton gradient is being created and oxidative phosphorylation cannot occur. So ATP production is stopped in its tracks. When this occurs, a cell can survive using a completely different metabolic process called fermentation. Fermentation, therefore, is a metabolic pathway that does two critically important things for that cell. Fermentation, number one, is going to allow all of that NADH to go back into the NAD plus form. That's number one. And number two, fermentation is going to allow glycolysis to continue producing small amounts of ATP with substrate level phosphorylation. Let's look at the first feature of fermentation. We said fermentation pathways are going to allow cells to regenerate NAD plus from NADH. That's important because we need glycolysis to keep going on in these cells so that ATP can keep being made. Even though it's not a very efficient way to make ATP, substrate level phosphorylation can still go on and it can still produce at least a small amount of ATP to keep the cell alive. In order for glycolysis to occur, though, we need NAD plus around to accept the electrons that are being released from glucose. So we need to regenerate NAD plus. And this is how it happens. The glucose molecule is going to continue to go through the catabolic process of glycolysis, producing pyruvate. And remember, that process of glycolysis makes a net gain of a small amount of ATP through substrate level phosphorylation. It also loads up NAD plus with electrons, creating NADH. Now, normally this pyruvate would go on into pyruvate processing and the rest of cellular respiration. But instead, it's going to be shunted into this different metabolic pathway called fermentation. And that's what they're showing us here. The pyruvate is going to be converted into some fermentation byproduct. And by doing that, the NADH will be relieved of its electrons. In other words, it will be oxidized and regenerate NAD+. Now, these fermentation byproducts really vary. It depends on the type of organism that is going through fermentation. In other words, there are different types of fermentation. The byproducts of fermentation tend to fall into one of two groups. They are typically acid products or alcohol products. In addition, some fermentation pathways also liberate gases. So fermentation occurs when pyruvate, rather than oxygen or another final electron acceptor, accepts electrons from NADH, allowing NADH to be converted back to NAD+. And as long as NAD+ is around, glycolysis can continue. And as long as glycolysis can continue, a little bit of ATP will be produced through substrate level phosphorylation, and that will keep that cell alive. There are many fermentation pathways, but the two most common forms of fermentation are lactic acid fermentation and ethanol fermentation. To summarize lactic acid fermentation, and this is the type of fermentation that humans perform and other animals perform when oxygen is not available to them. In lactic acid fermentation, pyruvate, produced through glycolysis, is going to accept electrons from NADH, so pyruvate will be reduced, and the product that pyruvate will be converted into is lactic acid, or lactate. And along the way, NAD+ will be regenerated. During alcohol fermentation, the pyruvate that's produced through glycolysis will be converted to a different product. The first product that is produced is called acetaldehyde. Acetaldehyde gets produced and a little bit of gas is liberated, and it's carbon dioxide. Then the acetaldehyde accepts electrons from NADH, gets reduced in other words, and ethanol is the final product. Ethanol is produced as an alcohol, and NAD+ is regenerated. On this slide, we can see the details of lactic acid fermentation drawn out for us.